This protocol allows the inoculation of viral vectors directly into maize plants that can be readily applied in most lab settings without the need for any expensive specialized equipment. Direct inoculation eliminates the use of an alternative host as an inoculum source, saving both time and resources. This method could be applied to additional viral vectors, maize lines, and potentially other monocot species. Begin by planting 1 to 2 maize seeds in the peat-based growing medium in small inserts placed inside trays. Place the trays in a growth chamber under 16-hour days at 25 degrees Celsius and 8-hour nights at 22 to 25 degrees Celsius, or in a greenhouse at 22 to 25 degrees Celsius with 16-hour days and 8-hour nights. Regularly water plants and fertilize once a week with 15-5-15 liquid fertilizer at 330 parts per million concentration. Prepare agrobacterium for injection by inoculating the agrobacterium strain containing the desired viral construct in LB media with antibiotics. Then incubate the flask or tubes at 28 degrees Celsius while shaking at 225 RPM for 24 hours. On the next day, pellet the bacteria by centrifugation at 4,000 times G for 10 minutes at room temperature, and discard the supernatant. Then wash the pellet with 1 milliliter of deionized water. Resuspend the pellet in 1 milliliter of 10 millimolar magnesium sulfate solution, and measure the optical density.